Hi, I'm Emily.
And I'm Laura Ann, and the title of our project is The Effects of Methyltransferase on the Phenotype of Transformed Escherichia coli.
So the basis of our experiment was we want to test how methylation affected the phenotypes of E. coli transformed with a plasmid. So what we did was we methylated a plasmid. We will explain that process soon. But we methylated the plasmid and transformed it into E. coli. We then cultured the E. coli on plates and then observed their phenotypes. The basic field of this study was epigenetics. Epigenetics is the study of heritable changes that are not from the DNA sequences. So if you see the two lines up here, these are actually genetically identical, but because of epigenetics, they look completely different. Eventually, epigenetics could be used to reduce the risk of diseases related to chromosome disorders and other things.
One mechanism of epigenetics is called methylation. Methylation is a naturally occurring process in most living cells, and it can cause gene silencing. So the way this works is that a methyl group, CH3, is added to a nucleotide in the DNA sequence. And this can prevent that DNA sequence from being read by the DNA polymerase, and therefore it's not transcribed and that gene isn't expressed.
So the protein that methylates the DNA is called a methyltransferase protein. And the type of methyltransferase that we use is methyltransferase. It's naturally occurring and is coded for by the DAM gene. It sequences on the DNA sequence GATC on the adenine. If you can see the methyl group in red there. And this has been discovered for a while. The sequence that methylates on was discovered in 1978.
So, plasmids. Plasmids are rings of DNA that are separate from the chromosomal DNA in bacterial cells. If one bacterium has a specific plasmid that encodes for a beneficial gene, it can replicate that plasmid and then share it with other bacteria so that those other bacteria can have that same gene. The plasmid that we used for this experiment to transform, should we? Technical difficulties, we apologize. All right, guys, we're back and still talking about plasmids. So the plasmid that we chose for this experiment was PBR322. And the reason we chose this plasmid is because it contains the gene for ampicillin resistance in E. coli. And this gene was sequenced by a guy named Sutcliffe in 1978. And he also found that this gene contains 6 GATC sites that can potentially be methylated by DAM methylase. Another reason we chose PBR322 is because it has multiple restriction sites that can be blocked by DAM methylation. So once that methyl group is put on top of this restriction site, the restriction enzyme can't come along and cut the DNA there because it's been blocked. So those were a few reasons we chose that.
So the type of bacteria that we used for this experiment was E. coli. We used them because they're a well-studied model organism. So we know the genotype and what their normal behaviors are. So if there's a change, we can easily find what caused that. Another reason we use this bacteria is because the strain that we use, DH5-alpha, naturally uses DAM methylation. The bacteria that we used were also chemically competent, which means that there were holes punched into their cell membranes using calcium ions, I believe, to allow DNA to travel in and out.
So the antibiotic that we used, again, was ampicillin. Ampicillin is a common antibiotic. It's used to treat lots of infections. It's a member of the penicillin family. And it kills bacteria by breaking down their cell membranes. So normal DH5-alpha E. coli cannot survive in the presence of ampicillin. It is killed. But when the E. coli has the plasmid in it, the plasmid PBR322 with ampicillin resistance gene, it can live on plates with ampicillin. So that was really helpful to our experiment in terms of making sure, first of all, that we transform the bacteria correctly and seeing the effects of methylation on that gene in the plasmid.
So our hypothesis for this was that the bacteria transformed with the methylated plasmid would not be able to grow on the ampicillin plates because the gene for ampicillin resistance would be blocked by the methylation. So one of the first things that we did once we started this experiment was we made ampicillin plates using LB agar and an ampicillin stock that we made. The stock solution was made by adding 1 gram of ampicillin powder to 10 milliliters of deionized water. And then we filtered that using a really, really tiny filter to make sure it was completely pure. We then added different amounts to the plates to make different concentrations. We had 5 different concentrations of ampicillin plates. We had 100, 50, 25, 12.5, and 0 micrograms per milliliter. And we wanted a variety of concentrations because depending on where the methyl groups are, it can affect the expression of the gene a different amount.
So for methylation, the first thing we did was we diluted a molecule called S-adenosylmethionine. Adenosylmethionine, also called SAM. And what this molecule does and why it's important to methylation is that it is the methyl group donor. And so if you've got the SAM molecule, I wish we had a diagram of this, but if you've got the SAM molecule, methyltransferase takes a methyl group from SAM and then puts it on top of the nucleotide in the sequence. And so first we diluted the SAM, and then we added water, methyltransferase buffer, SAM, our plasmid, PBr322, and methyltransferase in order into a microcentrifuge tube. We incubated that tube at 37 degrees Celsius for 1 hour, and then we stopped the reaction by heating it at 65 degrees Celsius for 20 minutes, according to the New England Biolabs protocol that we found online, because we got all of this stuff from New England Biolabs, so we figured we should follow their instructions.
So after we methylated the plasmid, we decided that we wanted to have 3 different types of bacteria. We wanted to have a control group with no plasmid in it, another group with a plasmid that was not methylated, and a third group with a methylated plasmid in it. So to transform the plasmids into the E. coli, we heat shock them. So to do that, you add the plasmid DNA to the tubes containing 50 microliters of the E. coli and incubate those on ice for about 15 minutes. You then put them into a 42 degrees Celsius water bath for 45 seconds, then replace them back on ice. This pushes the bacteria past its limits to adapt to changes in temperature, which makes it easier for DNA to pass through the cell membrane into the cell. After this, we added 1 milliliter of liquid media, in this case LB broth, and then incubated them at 37 degrees for 1 hour while shaking at 225 RPM.
So the next thing we did was culturing our bacteria. We made 4 plates of each concentration for each different condition: the bacteria with methylated plasmid, bacteria with unmethylated plasmid, and the bacteria with no plasmid. And so that was 60 plates total, which we poured by hand ourselves. So we put 20 microliters of E. coli onto each plate with a micropipette, spread it around with a sterile inoculating loop, and then put those plates into the incubator to incubate overnight at 37 degrees Celsius. We also made a liquid culture, and we did that by adding 40 microliters of E. coli of each type to 3 different containers, and each container had 50 milliliters of liquid LB, and they were all at an ampicillin concentration of 50 micrograms per milliliter. Sorry. And we did twice as much just because it was twice as much of a volume of LB in there. And we also incubated those. And the reason we made a liquid culture was so that we could run what's called an optical density test.
So optical density is basically the measurement of how much bacteria is in a liquid culture. So to find this, we took test tubes and filled them with about 10 milliliters of the culture and put them into that. That is a spectrophotometer, which is kind of the same thing that Moma was using. We're just using it for a different purpose. So we ran a blank of just plain LB media through there, and then we tested 1 tube each of the different types of E. coli.
So these are results of the optical density test. We did the test at 2 different wavelengths, 600 nanometers and 550 nanometers. And as you can see, there wasn't much of a difference at either wavelength between the bacteria with the methylated plasmid and the bacteria with the unmethylated plasmid. And at no plasmid, there was very little absorbance at all, which makes perfect sense because that's the media that no bacteria should have grown in since they didn't have the plasmid and there was ampicillin. So the optical density at both frequencies, the M- bacteria was slightly more absorbent, which would actually support our hypothesis that being unmethylated allows more to be ampicillin resistant, but we don't really know if it's significant because we didn't have a big enough sample size. So it could be due to factors other than the methylation of the plasmid as well.
So these are some of the plates that we made. And if you look up here, you can see that this is the methylated group and this is the unmethylated group. And as the concentration of ampicillin goes up, you can see that they both still grow about the same, even though the amount of bacteria on the plate decreases slightly. If you look at the bottom row, these are the control group with no plasmid in them. They only grew on the plate with no ampicillin in it. They did not grow at all on any of these. And here's a simplified chart of that showing where bacteria grew and where bacteria did not grow. And finally, these are 4 plates of the highest concentration for each type of E. coli. As you can see, these plates, the methylated and unmethylated groups, grew very well, and the bacteria with no plasmid did not grow at all.
So, our discussion. If more time were available, we would have liked to get quantifiable results, some quantitative results, just numbers, instead of qualitative, just observations. And one way that we could have done this was by growing our cultures in a way that would have allowed us to have countable colonies of bacteria rather than just lawns. And we could have done this by using a dilution series, and we could have streaked our plates differently. In order to get verifiable results as well, we would have liked to verify that the plasmid was actually successfully methylated, and we could have done this with bisulfite sequencing or by using those restriction enzymes that we mentioned before, specifically BSABI. It's one of the sites. And then a plasmid with a different gene might have been more easily tested. A huge amount of our research time, which we had a huge amount of research time, was spent actually looking for the correct plasmid to use, one that would have the right genes and the right properties to give us results that were understandable. If we'd had a little more time and gone a little more into that, we might have chosen a plasmid with different restriction sites or perhaps with a lac operon, which would have allowed us to do testing in terms of the bacteria's ability to metabolize lactose.
So not everything went exactly according to plan. Like Scott said, we are his Giggle monsters, so any experiment never goes exactly as planned, and we are amateurs. And we completely built this experiment up from scratch. We started with nothing, having no idea what kind of experiment we want to do, only that we were both kind of interested in epigenetics and methylation. So planning out this entire experiment took a really long time, which if we had another week, we could have gotten so many more results. We just need one more week. Yeah. But another mistake that happened was that we kept our E. coli at too high of a temperature. We stored them at minus 20 Celsius instead of minus 80. Yeah, it's really hot in there. But that greatly decreased the confidence by allowing the pores in the membrane to close up. And another thing that could have gone better was that only one of our transformations gave us methylated transformed E. coli that could be counted and give us good results.
So in conclusion, there was no observable change in the phenotype of the E. coli once it was methylated. And this could be due to many factors. There's the potential that it wasn't actually methylated at all. The bacteria might have unmethylated the plasmid after transformation, which is something that bacteria can do. It methylates and unmethylates as it pleases, honestly. The methylation possibly just did not inhibit the transcription of the gene. Our hypothesis might have been wrong. And so, we don't really have any conclusive data as to why there was no change. We just know that there wasn't. At least we didn't have big enough sample sizes for us to tell if there really was.
So before we go, we have some thank yous. I want to thank Excel Energy for sponsoring our research here and the Adolph Coors Foundation for allowing me to be at FSI today.
I would like to thank the Twinton Family Foundation for funding my time here at FSI, as well as the Edward Madigan Foundation for sponsoring my research. I am really, really grateful.
We also want to thank Lori Ball for making FSI possible and Scott Abella for teaching us about what it's like to be doing research. As you can see in that picture up there, that's at 1 in the morning after we got back from a 7-hour-long time in the lab. So thank you for teaching us all about that and for helping us do this experiment. We had a great time.
We'd also like to thank UNC for hosting all of us here for 6 weeks, as well as the rest of the FSI 2013 staff, the teachers and RAs and everybody who's really helped us out a lot this summer, as well as all of our friends who pulled all-nighters with us and have been really supportive during these past few weeks for sure. Thank you.